Okay, so here's white, red, and then here's W, W. Okay, so that's what a dominant phenotype looks like. You can either have a homozygous red, where it's RR, or heterozygous RW, but the red overwhelms the white. Now, so that's dominance. Now we have something called incomplete dominance here. And again, we start with the same parents. Parents are WW on the left, RR on the right. They're same thing, but instead of being all red, now it's kind of like blending. So it's all pink during the F2 generation. Okay? And you can see who these F2 are. They're WR, 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 WR. They're all heterozygous. Okay? So if we have, as we see here, have an R and a W, and here we have an R and a W. When you have RR homozygous, that's red. So this one is R, R. This one is pink, RW, WR, and then WW. So, oh, by the way, you can RW and WR are the same, right? So with pink, you have heterozygous, you have one R and one W for white. Okay? That's incomplete dominance. Now, why is this doing this? Oh, for Christ's sakes. Okay. Anyway, now on the right, up at the top, okay, let's see. That's dominant, and that's recessive. This is incomplete dominance like before, but here, this is called codominance. So you still get white and red in the flower. Complete red's not any washed out, no pink. Okay? You just get a white. So some of the cells are expressed as white and some are expressed as red pigment. And that's called codominance. So, and you can read this on, you know, the thing, 4 o'clock plants and the ratio that you should get. So here we have a red allele and a white allele. And here we have a white allele and a red allele. Okay? So if we go red to red, we'll end up with homozygous red. Okay? So it'll be R R. Now, if this one is red, and then over here is white, it will still be red, but it will be R W. Here we have a white. Okay, so same thing. If we're dominant, A1, you get the dominant phenotype, homozygous dominant, and you also get the homozygous dominant, heterozygous dominant with the dominant phenotype as a heterozygote. And then only the homozygote is called a recessive phenotype. So A2 means that, let's say like with the flowers, it's all white. A recessive allele, by the way, is not associated with disease. It's just expression patterns. Nor is a dominant allele have anything to do with how common it is. Yes?
So is that why there's codominance? Because it's not recessive, like one of the two
No,
isn't
it's
recessive?
dominant. Codominant is when in the phenotype you see both red and white at the same time, like with that flower. Dominant, you see all red. Recessive, you see all white. Okay? And then incomplete dominance, where you have A1, A2, you would have in between, like that pink. So the dominant allele has nothing to do with how common it is or how rare it is. It's an expression pattern. Nor does it have anything to do necessarily with, a disease. It's just an expression pattern. And these are examples of traits. I already went over this. Tongue rolling, freckles, dimples, widow's peak, detached earlobes are all dominant. These others are recessive traits. Okay, so this is, we're back to sickle cell trait. So if you have a heterozygous genotype, HBA, which is the normal allele, HBS, which is the sickling allele, half your gametes will have HBA and half will have HBS. Oh, so this is, we're going to start doing what's called a Punnett square. It's kind of like this. This is just all the different variations that you can find. Okay, let's predict the outcomes of something called a Punnett square. Very simple. Now, if you remember, there's two alleles, HBA and HBS. Okay? We have two people who are heterozygous. That means we say, let's say up here, it's the man, and he is HBA and HBS. And here's the woman. And she is HBA and HBS. Now, in the first square, we have an HBA, okay, and an HBA. So it's homozygous, HBA. This is HBS over here, so we have HBA, HBS. Here we have HBS, HBA, and here we have HBS. HBS. So, a child who has sickle cell anemia, that means they will be homozygous for the recessive allele, which is HBS. The only thing that where actually we get this is this square here. So that means we have a 25% chance that a child born to this couple will have sickle cell anemia. 0.25. Okay? Okay. So this is a Punnett square. Along one side, we'll say the man is up here, the woman is down there, okay? And all you do is you line things up, and you go HBA, H. Okay. And then here we have HBA, HBS, so it's a heterozygote. We'll say A and S. And here, S and A, and then S and S. So what's the probability of having a baby that has sickle cell anemia?
25%.
Right. So, okay. Now, what's the probability that there are carriers? The parents are carriers, or the children are carriers of the allele? 50. Right. You have up on the right, top right, 25%, and on the bottom left, you have 25%. Those are both heterozygotes. Okay. Now, there actually, in this case, with the sickle cell trait, is there's not dominance or recessiveness. There's a bunch of different things. So, on the left, you have, actually, and it shouldn't be dominant, it's homozygous. Okay, so it's homozygous HBA, HBA. So we'll say A. Okay, and that's now with malaria in the area, that means AA is low fitness because it has no protection against malaria. So the person in that area with that phenotype or genotype and then phenotype will be susceptible to malaria. Okay. Now, in this one over here, let's take a look. This is the same thing. If there's no malaria present, it has the highest fitness. Okay. Now, if you have HBS, you have sickle cell anemia, you have sickled red blood cells, it's lethal, but you need both copies of S for this to happen. Okay, so you can see what it looks like. So these cells, blood cells, it's called anemia because they don't carry oxygen very well. And in a low oxygen environment, they also start to sickle, and then they tend to clump together, especially in the capillaries and all that. So in the past, most people that had sickle cell anemia, they died before they could really breed. There's some treatments now, but nothing to cure it. So it's a pretty heavy cost to pay, and it's only paid in areas where malaria is endemic. And so you lose 25% of your offspring, essentially, or whatever it is. A quarter of all the people up there in the area who will have that sickling cell anemia, and they'll be wiped out in terms of the breeding pool. So that means that malaria is killing more than 25% of those people in the population. Okay? Otherwise, selection wouldn't favor this. It'd be too heavy a cost. Now, in the middle here, we've got HBA, HBS, that's a heterozygote. Well, in the past, we would say that, oh, it was dominant, and that means you ended up with mostly normal red blood cells, but that's not really the case. It's actually, in terms of the phenotype, if you look closely, okay. What I'll do is, when I post this, I'll go back and explain that because I didn't record that. Okay. Okay. This is actually where HBA, HBS, that's called sickle cell trait. Not sickle cell disease, it's sickle cell trait, which means that the red blood cells are resistant to being infected by malaria. Okay, so sickle cell disease, you actually, to get it, you need both copies, HBS, HBS, cystic fibrosis, another genetic disease like that, is also recessive. You need both copies of the allele. Huntington's disease is a dominant genetic mutation. That means all you need is one copy and you will express the phenotype. In fact, with a dominant disease like that, if you ended up, let's say, both parents had Huntington's disease, they had a child, the ones that inherited two copies, that I'm sure that fetus would never develop normally and would actually be born alive. Okay? So Huntington's disease, now there's another thing we're going to learn about it, but it's still around mainly as a dominant disease, even though you'd expect selection to wipe it out, because it doesn't show up until adulthood, later in life. Okay, so here's cystic fibrosis, same sort of thing. Okay. So the CFTR protein, the normal protein, actually transport chloride ions across the cells of the lung. Now, and then we can actually look at that. Okay. So here's the chloride ions. Here's the normal CFTR channel, right? And the chloride atoms come into the cell. Now, the trouble is that the channel does not move chloride ions, so the mucus, in the way it is expressed, is very thick and viscous on the outside of the cell. And those individuals who have that very thick mucus, first of all, it builds up in the lung cells, and so they can't breathe, and they're also much more susceptible to other infections. Now, some of this is also expressed in the intestine, and so there's also intestinal problems with this disease as well. As I mentioned, it's a recessive. You need 2 copies of the funky allele. Okay, now it's been hypothesized that this genetic disease is actually much like sickle cell anemia, which is to protect against malaria. Well, it's thought that this protects against typhoid fever. Okay? Now, there's not much typhoid fever left in the world, but there is some. So you're still paying a high cost for having 2 copies, but you're also getting a benefit in terms of being resistant to typhoid fever. Okay. So, how many CFTR alleles are needed for the person to be a carrier? 1 or 2? 1. Okay. To have, in terms of having cystic fibrosis, how many alleles do they need? Anybody? 1, 10, 2? 2. Okay. So this is kind of what it looks like, as before. The woman, so if 2 carriers marry, what's the probability that the child will have the disease? Same as 25%, just like sickle cell anemia. Okay, so now though, if a man with CF marries an unaffected woman, okay, whose mother had cystic fibrosis, in other words, her mother had 2 copies of the bad allele, okay, what is the probability that the child will have the disease? Okay. Now, remember, the mother, the woman's mother, but the father, he probably has a normal copy. Okay? So that would make her, if you see on the left, heterozygous. 1 normal copy, 1 mutant copy. And then there's this man who actually is cystic fibrosis. In other words, he has 2 copies of the mutant gene. So then you just go across. Here's the bad allele right here, the mutant, and the mutant. And so you both have mutants here. Oops. Here you also have mutant. Here you have heterozygote. And here you have heterozygote. So the child has a 50% chance of inheriting 2 mutant copies. And it's only because the man has 2 copies and the mother has 1. Okay. Now, Huntington's disease is caused by a very rare dominant allele. And it causes a protein that's non-functional that has to do with neurological signals in the brain. So there's people who express it have these terrible neurological symptoms, and they usually die fairly rapidly. However, it doesn't really show up until the person is like, 45 years old. So now, if it showed up, let's say when the person was 10 years old, it would be wiped out. Selection would wipe it out, right? Because before the person bred, they would be dead. So if your father has just been diagnosed with Huntington's disease, a dominant condition, what are the chances that you will eventually develop the disease? Okay, so we can do a Punnett square. Actually, hold on. Okay, we'll say the man's on top. He's got 1 copy of Huntington's and 1 normal. And then the mother has 2 copies of normal. Okay. So if you have normal plus Huntington's, it's a dominant trait, right? It shows up as in the heterozygote as well as the homozygote. They've got Huntington's disease. Okay? And these are normals. And then same thing here. NH, got Huntington's. And then here, NN. So essentially, the probability is 50%. If your dad has Huntington's disease, or your mom has Huntington's disease, it means that there's a 50% chance that all the offspring will have the disease. They may not show the disease until later in life, but that's their chances. Now, you probably, I don't know if you know who Woody Guthrie is, but he wrote this song, "This land is your land, this land is my land," you know, from California to the New York skyline. Okay. Anyway, he's great. He was great. Let's see. One of the lines was, and as I was walking, I saw a sign that said "No trespassing," but on the other side it didn't say nothing. So this land was made for you and me. You know, the hell with trespassing. So anyway, it ran in his family. Everybody, there was this certain number of people in his family who would, you know, they'd start showing Huntington's at 40 or 45. And it not only had a condition where, you know, kind of spastic movements, but also it had personality consequences. So people tended to hallucinate as well. And eventually he developed it. He actually lived with it for like 14 years, so it wasn't really strongly expressed. The Guthrie family has been a long-time musical family, and his son Arlo, he had this album about Alice's Restaurant, if you ever want to look it up. It's very funny. Anyway, no one knew whether he inherited a copy of the allele or not. Turns out he didn't. but you really don't know unless, of course, you sequence the person. And then if it comes out that, oh, you have it, then you have to live with that until you start falling apart. So this is what we just did. So on on the left, we have a woman who's normal. She's got the normal copy, which is a small h. So she's HH. And then we have a man who's actually heterozygote. But remember, it's a dominant trait, so all you need is one copy. So he's got a large H, which is the abnormal allele, and then a small h. And we can now start filling things in. So if we take the woman who's got a small h at the top, and the man who's got a large H, that's 25%, and the same thing on the bottom, H and H large H, that's 25%. That means they both have a large allele, large H, which means that they will develop Huntington's disease. Now, of course, let's go off on the top again. The woman is small h. The man is also small h on the far right. So HH, that's 25%. And the same thing on the bottom. H for the woman and H for the man, HH. And so in the end, you end up with a dominant condition, right? So it's 50%. That you'll end up, 50% of the children will end up with Huntington's disease on average. Doesn't necessarily mean, it's kind of like flipping a coin. Sometimes you might flip it, let's say, 2 times in a row. It's Huntington's, and only the third time, it's the other one. So that'd be 3/4, right? So, but on average, it'd be 50%. If a trait, that means the phenotype, skips a generation, is that allele dominant or recessive? Okay? Okay, so here's the normal wild type, white mother and father. Here's the individual who's recessive. Let's say mother has the allele, father doesn't. If this individual donates the allele, they'll have it a certain amount of the time in offspring. Okay? Now, no one in this second generation will actually express the phenotype. In other words, the next generation, it will skip. You won't see example of the phenotype. It's only in the third generation where you have an individual, 2 individuals that actually have the allele, and they'll produce offspring that are blue. So in terms of skipping the generation, it only happens with recessive alleles. Okay, so mistakes in meiosis, non-disjunction. So non-disjunction, failure of chromosomes to separate during cell division, results in gametes with 1 too many or 1 too few chromosomes. Most embryos that result from such gametes will die before birth, but occasionally they won't, but they will certainly have a mutation that expresses a phenotype, and that phenotype will probably have a lower fitness than a normal individual. mistakes in meiosis, here we are. Non-disjunction, of course. Events are viable in some cases. So like trisomy 21 produces Down syndrome. In terms of being expressed and how much it might disable the individual, it varies. There's some very high-functioning Down syndrome people. They all have a certain kind of face that you can easily recognize. So here's 21 down here. And during meiosis, he ended up with 3 copies, okay? An extra one. Okay, so now, if it had just been 1 copy instead of 2, it would have been lethal right off the bat. So autosomes are the same thing as, let's see, they're the same thing as somatic cells. Okay? So we have autosomes, 22 pairs of homologous chromosomes that are autosomes. Those are never donated during sexual reproduction. And then we have 2 sex chromosomes, an X and a Y. And this is what a female looks like, XX, and a male has an X and a Y. Okay. Now, where does the male get his Y chromosome from? The father or the mother? Father. A male is always XY. If the father donated an X, you'd end up with a daughter. So mother is XX, father is XY. You would then have a 50% chance of being male or female, depending on whether you inherited an X or a Y. There's different, because of this, there's different expression pattern. There's a whole developmental pathway during fetal development that turns an embryo male. That pathway is actually governed on a loci or a number of loci on the Y chromosome. Okay. Now, there's some other things that can happen. Other mistakes in terms of non-disjunction. Okay. Let's see. Okay. Now, a female, remember, the genotype without a Y chromosome will exhibit feminine characteristics. Always. So even if you end up with an individual who's only has 1 copy of the X, that individual would exhibit female characteristics, but XO is probably not viable. But some individuals are triple X, and they would exhibit as female. So, it's not, so it's, the syndrome is normal, Turner syndrome, and triple X. Turner syndrome is associated with a bunch of disease phenotype or pathological phenotypes expression. Okay? Now, if a male has an XY, he's normal. If he inherits two X's and a Y, he's got another genetic syndrome or disease called Klinefelter. Turner and Klinefelter both exhibit retardation and a bunch of different kind of pathologies. And then, of course, you can actually have an XYY, kind of like a super male. There was a time, quite a number of years ago, that suggested that males with an extra copy of Y were more aggressive and were more likely to commit crimes. Now, this is before they actually were sequencing, but they had figured it out using Mendelian genetics. Because they could actually see the chromosomes. They didn't know which allele it was, okay? So there was this whole, should we test people, males, for having an extra Y? You don't have to worry about the ones with two X's and a Y because they'll, be kind of like girly men, like what was it? Arnold Schwarzenegger said. Yeah. So, well, it turns out it was all BS. They didn't know what they were talking about. But there are certain phenotypes that are associated with that. So this is more disjunction stuff like Down syndrome. Okay. So in terms of genetics and then expression patterns, it's different in terms of sex chromosomes. Now, oftentimes, let's say back to flowers. In incomplete dominance, let's say with a red flower and a white flower, you'll get pink. That's called haploinsufficiency. Not enough of the red pigment, which is a protein, is actually produced. Because the person has one normal copy, and then one red copy. Okay? Well, with sex chromosomes, it's exactly the same thing. Now, the females are fine. A female has two X's, right? And so, now, you don't always get haploinsufficiency. A lot of times you get enough protein made that you're still fine. Let's say you have a mutation in the X chromosome, and you're female, there's going to be a rescue effect from the other chromosome with the normal mutation. So there won't be haploinsufficiency. Now, the trouble is, if you're a male, that means you are the heterogametic sex, and you are XY, okay? Okay. That means if you have a problem on the X, you will not get a rescue effect from the Y, because the Y doesn't have that gene on it. Okay? So it means that males, or of course if you have a mutation on the Y, well, that's the male, and it'll show kind of a dominant expression pattern. And many of the genes on the X chromosome, of course, are have to do with reproduction, including the amount of sperm you make, the quality of the sperm you make, all sorts of different things. In fact, as I mentioned quite a while ago, there's Neanderthals and modern humans interbred 40,000 years ago, and then in the East Asia, again, a little bit more recently. So there are people who have between 1% and 3% Neanderthal in them. However, if you actually look at the Y chromosome in the males, there is basically no Neanderthal Y chromosome there at all. It means that selection has selected against it. The genetic background produced individuals who were not fit, whether they were unviable or they were sterile. Okay? And it's not just the X chromosome, it's also the Y. I mean, it's not just the Y chromosome, it's also the X. So there were also genes on the X that affect male fitness as well, that were had no Neanderthal compared to the other somatic cells in a lot of places that were enriched with Neanderthal DNA. Okay, so now it's the heterogametic sex that expresses any mutations and shows a problem. Because an individual, a male whose XY, can have a mutation on the X chromosome, and the Y, of course, doesn't rescue it, or on the Y chromosome. So the heterogametic sex is much more likely to suffer from these problems than the homogametic sex, which is female. So they're XX. So a lot of times you'll have an XX female who has one copy of, let's say, a deleterious allele, and she mates, she's a carrier. She doesn't show any disease or anything like that. And she mates and they produce a son who actually has full expression. And we'll see this in a minute. So when you have loss of fitness because of being the heterogametic sex, this is called Haldane's rule. So it's usually the females. You're never going to show something bad unless they actually, let's say the male had that bad, obviously the bad X2, and he donates, and then the other mother has a copy of the bad X, and you end up with a daughter with two bad copies. But the chances of that happening are very, very small. So with the heterogametic sex, there's more likely to be viability and sterility problems than the homogametic sex. As an aside, females in birds, it's the opposite. They have a ZW sex chromosome, and it's the females who are ZW and the males who are ZZ. So it's the females that will exhibit any problems associated with the sex chromosomes. So who determines a baby's sex again? Dad. Okay. So if the dad donates a Y, the baby is male. If the dad donates an X, the baby is female. So males get their Y from who? Okay. And their X from mom. So if you're a boy and you have problems, you can blame it, you know which parent to blame it on. Okay? My mom gave me a bad X. My dad gave me a bad Y. Period. I mean, that's just the way it is, you know? Like with Huntington's disease, you have, well, first of all, it's not on sex chromosomes, so we can forget about that. Okay. And there's another thing. Can males be carriers of X-linked traits? That means a carrier doesn't express, let's say, a problem, whether it's male color blindness or something like that. So a carrier doesn't express it, so a male can't be a carrier. If he's got a problem on the X chromosome, he's going to express the problem. Only females can be a carrier. Okay. So, why do some genetic conditions affect only males more often than females? And of course, that's Haldane's rule. Male has a single X, so he has only one copy of these genes, not two. The female has two copies, so she can carry a recessive allele without showing the trait. So, in terms of the heterogametic sex, it acts just like a dominant allele. Okay? Because it's actually on the sex chromosome. With the female, it doesn't. Anybody in here colorblind? Can't see red and green? Okay. Now, it turns out color blindness is caused by a mutation on the X chromosome. And if a colorblind blind individual, man, donates an X, he can donate a bad X with a mutation, or the mother, though, is usually the carrier. She has bad X, but she has a rescue, so her daughters are fine. And this kind of Haldane's rule sort of thing, you really start to see it in royal families. As you probably know, or maybe you don't, wherever there's been royal families and there's consanguinity, which just means like royal blood, and not breeding with anyone else but someone with royal blood, whether it's with your sister or brother or your stepsister or, your half-sister or brother or your cousins, alleles that are recessive are more likely to be exposed. Because it turns out every one of you has about, probably 40 or 50 mutations that no one else has. They just happen. Most have no or little effect on you whatsoever. However, it could be that, in fact, if you had this mutation and there was someone else, because it's been passed around within kind of like a large royal family, the chances of getting 2 copies of it, it might not have no effect. It might actually have a very strong effect. So whether it's blue-green color blindness or it's hemophilia, the clotting factor, also insanity, the royals produce, it's amazing how the effects, I actually looked at a paper on the effects of inbreeding in royal families. And most of the kids, in fact, a lot of them were not very healthy. Very few of them, in a lot of cases, ever made it to adulthood. And if they did, they were worthless. So now it turns out, of course, if you keep doing this, eventually, over many generations, you'll weed out all those recessive alleles with the individuals with 2 copies of it. And so that mutant allele will start to disappear from the population. Okay, so let's take a look at this. If a colorblind man marries a woman who is a carrier for the recessive allele, what is the predicted outcome for their children? So these are 6 linked traits. Why don't we make a Punnett square? The colorblind man, we'll do in blue. He's got an X with the colorblind allele, and he's just got a Y. Okay? And then the woman is a carrier, so she has one X, that's colorblind, and one X, that's normal. Okay, so first of all, in the upper left quadrant, we have an X with color blindness for the woman. Okay, and the man also has a colorblind X, so we're basically colorblind. We have C, and this is actually a female who's going to be colorblind. Now this rarely happens, but it could, like in a royal family or something like that. Now, if we go across, we still got a colorblind female, and we've got a Y. So we'll have a man who is colorblind as well. Okay, now on the bottom here we have a normal X, and we have an X and an XC, so that's a carrier, and we also have a X and a Y. Okay, so let's take a look at this. We have a female here, child. We have a male child here that is, in fact, colorblind. On the bottom, we have an X and an X with a carrier, and so this is a carrier female. And here we have an X and a Y, normal male. Okay, so their children, we will have one female that is colorblind, a female carrier, a male that is colorblind, and a male that is normal. this again. No. Okay. Okay. What fraction of their sons? Okay, so here we have X, normal female. Have an, oh, this is also an X. No, that's female, that's right, and this is a male. Okay. And then we have this mutant X. Okay. And then we have a male that's normal male, X, Y. And then we have a mutant male, X, Y. So it asks, if you have a carrier female marries an unaffected male, so the male doesn't have recessive hemophilia, what fraction of their sons will be affected? So let's take a look. As we said, we've got the female on the left, she's got an X normal and an X mutant. And we have a male who's X normal, Y normal. If we go normal female, we've got two X's, a normal female. If we got a normal female and a normal male, XY, we also have a normal male, right? Okay. Now, we get to the bottom here. If we have an X, a mutant X from the female, and a regular X from the male, like here, we have a carrier female, X carry. Okay. And if we have an X mutant from the female and a normal Y, we then have a hemophilia son. Okay? Now, the question is, what fraction of their sons? Okay, so fraction of their sons. Not all their children. Remember, there's two female children. Just their sons. What fraction of their sons will be affected by having hemophilia? Well, there's only two boys here, right? One is normal, and then one has hemophilia from his mother, who was a carrier. So it's actually 50%. Okay? Simple. That's their sons. It's actually 1 out of 4, 25% for all their children. But they're only asking here their sons. Okay. So, if you are a colorblind male, which parent gave you your color blindness? Well, color blindness is on the X chromosome. Did you? Yeah. So you got your X chromosome from where? Your mom. You can't get it from your dad because you got your Y from your dad, and that's why you're male. So in other words, you can blame it on your mom. Now, of course, if it's a mutation on the Y chromosome, then we know you got it from your dad.